Great clinical research professionals aren't born, they're built. In each episode, hear from leading experts in clinical trial management as they inspire and motivate you to become an effective leader. Welcome to the Clinical Trial Podcast. Here to help you accelerate your career is your host, Kunal Sampat.
Hi, friends. Welcome to another episode of the Clinical Trial Podcast. I'm your host, Kunal Sampat, where my job is to help you accelerate your clinical research career. Cardiovascular disease is one of the leading causes of death around the world. It has a negative impact on human lifespan and health span. Because of the significance of cardiovascular disease, there are several cardiovascular clinical trials that are being conducted at any given time. To learn more about the important field of cardiovascular research, I invited the world's top cardiovascular researcher, Dr. Gregg Stone, on the podcast. Dr. Stone is an interventional cardiologist and director of academic affairs for the Mount Sinai Heart Health System and professor of medicine cardiology and professor of population health sciences and policy at the Icahn School of Medicine at Mount Sinai in New York. He has served as the national and international principal investigator for more than 150 national and international multicenter randomized clinical trials and studies, many of which have led to new device approvals or indications in the United States. He has authored more than 3,000 manuscripts and abstracts published in peer-reviewed literature and has delivered thousands of invited lectures around the world. With an H-index of 202, Dr. Stone has been recognized in Nature Medicine as one of the most prolific authors in science and by the Web of Science as among the top 0.01% of cited researchers in science. Dr. Stone's area of expertise include interventional therapies of acute coronary syndromes, myocardial infarction, and cardiogenic shock, drug-eluting stents and bioresorbable scaffolds, left main and complex coronary artery disease intervention, antiplatelet and antithrombotic pharmacotherapies, transcatheter valve repair and replacement, interventional arterial and pulmonary hypertension and heart failure therapies, left atrial appendage closure, intravascular imaging such as IVUS, OCT, and NERS, vulnerable plaque diagnosis and treatment, adjunctive interventional devices, saphenous vein graft therapies, contrast nephrotherapy, clinical trial design, and regulatory issues. Dr. Stone was the director of the Transcatheter Cardiovascular Therapeutics, TCT, from 1998 to 2021, the world's largest symposium devoted to interventional cardiology and vascular medicine. Dr. Stone founded the annual National Interventional Cardiology Fellows Course, Transcatheter Valve Therapies, TVT, and the Chronic Total Occlusion CTO Summit, and co-directs multiple other meetings in the United States, China, Russia, Europe, South Korea, and elsewhere. Prior to joining Mount Sinai Heart Health System in 2019, Dr. Stone was the professor of medicine and director of cardiovascular research and education for the Center for Interventional Vascular Therapy at Columbia University Medical Center and New York Presbyterian Hospital, New York, New York. Previous to this, Dr. Stone held similar positions at Lenox Hill Hospital, New York, and Washington Hospital in Washington, D.C., and is a past chairman of Cardiovascular Research Foundation. Previously, he was the director of interventional cardiology at Cardiovascular Institute at the El Camino Hospital and Stanford University Medical Center in California. Dr. Stone completed medical school at Johns Hopkins University Medical Center in Baltimore, Maryland, and his internship and residency at the New York Hospital Cornell Medical Center in New York City. He completed his general cardiology fellowship at Cedars-Sinai Medical Center in Los Angeles, California, and subsequently a dedicated fellowship in advanced coronary angioplasty in Kansas City, Missouri. Please join me in welcoming Dr. Gregg Stone on the Clinical Trial Podcast. Dr. Stone, thank you so much for accepting my invite and being on the podcast today. I'm really excited to talk to you about cardiology clinical trials, and you are the world expert on this topic. So thank you again, Dr. Stone.
My pleasure.
I want to maybe start off with discussing some of the most groundbreaking or practice-changing cardiology clinical trials that you've been part of or closely involved with. What have they been, and what made their findings so impactful? I know I was part of one of them, but I'm sure you've been through many more than one.
Well, thanks. I've been very fortunate to have been involved in a lot of important clinical trials. Important meaning that they had an impact on the way that physicians practice and the outcomes that it leads to for patients, basically either saving lives or improving quality of life. And probably the first one, and one of the most memorable, was the PAMI trial, the original primary angioplasty and myocardial infarction trial, which I was one of the co-principal investigators on, along with Bill O'Neill and Cindy Grimes, and participated in this very shortly after my fellowship. And it was really the first randomized trial of primary angioplasty versus thrombolytic therapy in patients with acute myocardial infarction. And at that point in time, thrombolytic therapy, also known as fibrinolytic therapy or clot busters, were the way that about 99.9% of all eligible heart attacks were being treated around the world. And there was a relatively small group of people led by Jeff Hartzler, and then the mantle was picked up by Bill O'Neill and Cindy and myself and Bruce Brody and others, who believed that mechanical reperfusion therapy at that time with balloon angioplasty would lead to better flow in the infarct artery, and by leading to a much larger lumen would lead to a lower rate of reinfarction, improved myocardial salvage, better quality of life, et cetera. And so at that time, though, that was pretty outrageous. The idea of taking patients to the cath lab, introducing all those delays, injecting dye, and otherwise manipulating the coronary arteries to somebody who was actually having an acute coronary occlusion, most people thought was crazy and would be dangerous. And it took a large number of pilot series to suggest that this could be very effective. But ultimately, it required the randomized trial. And that was the PAMI trial, which was the first one to show that death or reinfarction could be reduced by about 60% with primary angioplasty compared to fibrinolytic therapy in patients with ST segment elevation MI. But that wasn't the end of the story. It required 22 more randomized trials, which basically replicated those results. But ultimately, PAMI was proven to be right and was the standard bearer that led to the whole world ultimately changing over about a 10-year period to a reperfusion therapy being done with, primary angioplasty, with thrombolytic therapy now being rarely used only in patients who really present at very remote locations. So that was very, very exciting. And there's no doubt that over the, 30 plus years now since the PAMI trial, that has literally saved millions of lives.
Wow. And then after that, what was the next, like, was it drug-eluting stents or bare metal stents after that, or?
Well, I've done a lot of work in a lot of different areas, and a lot of the studies I've done have focused on acute myocardial infarction, but then also chronic coronary artery disease, both for patients with stable coronary syndromes, acute coronary syndromes, and then preventative therapies. In particular, most recently looking at vulnerable plaque detection. And that's the newest and I think latest paradigm shift that we're about to enter into. So along the way, we tried to make angioplasty better. And we went from balloon angioplasty to atherectomy devices, then ultimately to bare metal stents and to drug-eluting stents. And I was, very fortunate enough to be the principal investigator that led to, the U.S. approval of the Taxus drug-eluting stent, the U.S. approval of the Xience drug-eluting stent, the U.S. approval of the Promus and Promus Element drug-eluting stents. So again, it's been, my honor to really help usher those devices into the U.S., which have continued to improve outcomes. And then we also proved that those were safe and effective in patients with acute coronary syndromes and STEMI. So everything is a step, and all these things take many years to accomplish. We're now working. No, we're now working on the last remaining, issues in acute myocardial infarction, and that is to try to salvage more myocardium, which has been a large nut to crack, because a lot of patients present late, and there's reperfusion injury and distal atheroembolism and a lot of other things that make infarcts large, even if you can get the infarct artery open, and also to try to improve outcomes in cardiogenic shock, which is really the last, holy grail, really, where patients still have about a 50% 30-day mortality. So it really never ends. But I mean, when I try to think about how far we are along now compared to where we were, 35 years ago, it's really extraordinary.
How did you get that opportunity to be the co-investigator? Did one of your professors ask you to be the co-investigator, or how did that come to play?
Yeah, well, early on, when you come out of your fellowship, you're barely trained to be an independent operator, and you're barely trained to know what research is. So you have to start somewhere, and I give lectures and talks to interns, residents, fellows all the time on how to develop an academic career, which combines both patient care and clinical research. And early on in my career, I was no different than anybody else. And so, people knew that I was very interested in primary angioplasty from my fellowship with Jeff Hartzler. And so I developed a network of people and got to know, Bill O'Neill, got to know Cindy Grimes, Bruce Brody, and David Cox, the other people that were really involved early on in the field. And early on, there was this discussion about doing this randomized trial, and it was only done with a handful of investigators, just about 10 investigators. And again, I was fortunate enough to be in that circle and offered to volunteer my time because we really had no funding to do the study. I mean, this was well before industry funding. I mean, I consented all my own patients. The database for the PAMI trial was an Excel spreadsheet. It was a paper case report form. So it was nothing the way it looks like today, but it still was able to change the world, and in a very, collegial and collaborative environment. And that's the essence of, clinical research.
Thank you for sharing that story. So when it came to designing the trial itself, how did you learn how to design a clinical trial back like 35 years ago? Like what how did you what were there some resources that you accessed, or things that you kind of were curious about that led you to learn about those things?
Right. Well, it really starts with, the desire to want to answer a question. And actually, back when I was starting out, it was even debated whether we really needed randomized trials, or could an observational data be enough? Couldn't large single arm studies, as they're called now, provide you with enough convincing data of safety and effectiveness? But then there were a few early randomized trials that were done. The one that really sticks in my mind was the caveat trial, the original trial of balloon angioplasty versus directional atherectomy. Directional atherectomy, no doubt, led to much more beautiful angiographic results than balloon angioplasty. And almost everybody was convinced that that would lead to better clinical outcomes. But when the clinical trial was run, called Caveat, that was run by Eric Topol, actually, it had a higher rate of complications, in particular procedural myocardial infarction. And the overall outcomes were better, similar or better with balloon angioplasty. So no one saw that coming, and that led to us figuring out how to improve the results of first directional atherectomy. That was in the pre-stent days. And it showed the power of randomized trials. You have to have a control group, and it's got to be an equally balanced control group that doesn't have confounding elements in it, to be confident that the groups are matched, with the one exception being the therapy that you're testing. So then the question is, okay, if you start to believe that randomized trials are the true science to answer clinical questions, there's no doubt that they're very expensive, they're very time consuming, they're very complex, and you can't randomize everything, as people will say. Some things don't need to be randomized, as people will argue. You don't need to randomize parachutes, right? No one's going to do a randomized parachute of jumping, 25,000 feet out of a plane with or without a parachute. So you have to ask yourself, what's the clinical need? What is an important enough question to answer that warrants, going through all that effort and time and expense to organize a multi-center, clinical randomized trial to answer the question. And then once you decide that it is worth doing, then you obviously have to figure out where the funding is going to come from. Everything starts and ends with funding. No funding, no trial. But then once you've got funding lined up, then it starts to get really difficult. And designing the right clinical trial, asking the right question, knowing who the right patients are to enter, having the right study processes, selecting the right endpoints, powering the trial appropriately, collecting and analyzing the data accurately is, really the essence of doing it right. And I always, tell my fellows that not everyone knows this, but approximately 2/3 of all randomized trials are negative. 2/3 are negative after all that work and all that time. And I say of the ones that are negative, I think about half of the ones are negative because the device or the drug or the therapy just doesn't work. And you didn't know it until you did the trial. But I think the other half are probably negative because the trial was run poorly. You had the wrong patient population or the wrong endpoint, or it was too small, or there was too much crossover, or hundreds of reasons that a trial can be negative, even if the therapy truly is safe and effective. So, that's what I've, dedicated my career to try to overcoming, to get rid of those 1/3 of false negative trials, to try to run trials that are robust enough and well done enough that the trial doesn't get in the way of proving a device or a drug is safe and effective. It's hard enough to do that without us getting in the way of the effort.
Yeah, thank you for sharing. I read a book recently, and I'll put it in the show notes for people on designing clinical research. It's been written by UCSF, set of UCSF professors, and it was really amazing to kind of go through the different trial designs and understanding how to think about trial design.
Yeah.
So, from your perspective, what are the biggest challenges in designing and conducting high quality cardiovascular clinical trials?
Well, again, it goes down to, paying attention and putting in the energy and time and detail that it requires to every aspect of the clinical trial stage. There's really no shortcuts, and that's the problem, because really good clinical trials are very expensive, and they take a large team of people to both design and carry out. It's not just one or two people that can run an effective clinical trial today, especially in the complex, structures that we're working in, where we want trials to be generalizable. So we have to, almost all important clinical trials are multicenter, usually with dozens, if not 100, clinical trial centers. Each clinical trial center having numerous operators. They all need to be trained. You have to choose the sites that can effectively enroll patients, have the right patient population, and then they have the right infrastructure of research and a clinical track record to be able to accurately enter data, to be able to consent patients appropriately, to keep and maintain their records, follow all regulatory guidelines, et cetera. Um, you obviously need a sponsor, which is usually either industry or government. And there's pros and cons of working with each. And then industry or government will then usually partner with an academic core group, usually one or two principal investigators, and then a steering committee that will help design the trial. Then you put in operations teams to help carry out the trial. Large trials are complex. Usually, there'll be an outside contract research organization that'll be hired to help either manage the sites or monitor the data, collect the data, train the sites, et cetera. There'll often be an academic research organization, for example, a university or private-based academic group that partners with the sponsor on the trial as well. And of course, FDA is a key part of all of this. For any investigational device or an approved device for which you're testing an unapproved indication, that all has to go through FDA. So you've got to be very, very comfortable and knowledgeable about working with FDA. And even for studies that don't need to go through FDA, of course, they all go through their local institutional review boards. Many of these studies are often not just US based, they're often multinational. So then it gets much more complicated because either you or somebody has to have relationships overseas and be familiar with or know how to qualify good research sites, whether it's going to be in Europe or South America, or Australia, New Zealand, or the Asia Pacific region, or Canada, or Mexico, or other areas. So it can all be, quite complicated to be able to perform. Sorry, just give me one second. Okay. So, we designing the trial, really is a whole art form in itself. And again, you've got to, know what clinical question to ask. Basically, most clinical studies are designed to prove, the effectiveness and the safety of a therapy. It's usually either a device or a drug, or it can be a strategy. For example, PCI versus CABG. And you want to prove both effectiveness and often there's a parallel goal of improving safety as well. So sometimes you'll have an effectiveness endpoint, sometimes you'll have a safety endpoint. Those are superiority trials. Sometimes you'll run a non-inferiority trial and try to prove that a new therapy is at least as good as a prior therapy. Then maybe it has other advantages, but you want to prove non-inferiority, meaning at least as good or better. So you've got to ask the important clinical question, and there's nothing more important than that. And then you start drilling down. Well, what's the background? What's the baseline knowledge base of how patients are treated with a particular condition that you're studying? What are the standard of care treatments? What's the background of the state of understanding of the device or drug or therapy that I'm treating to suggest that it may safely improve outcomes? Are we ready to do a clinical trial with that new approach? Then who are the right patients? And often, you'll have, 6 to 10 inclusion criteria, all of which must be present. And often we'll have 10 to 30 or more exclusion criteria. So we're often drilling down on a very, very specific patient population to try to isolate the effect of the therapy. So for example, we'll often exclude patients who are on dialysis. They might have renal insufficiency because patients on dialysis have so many complications, so many adverse events, and their pathophysiology is often so different than patients not on dialysis that just including 1 or 2% of patients on dialysis into your study could skew the results of the study. And you might come up with 10 or 20 different exclusion criteria like that. Well, maybe patients with bleeding diatheses, or patients that will refuse transfusions, or patients who are unable to tolerate antiplatelet agents. You can go on and on and on. And then, once you have your exact patient population, you have to choose, well, what are the endpoints? What are we trying to show? And that's a whole art form in itself too. Obviously, mortality, or the flip side of survival, is the ultimate endpoint. But most of our conditions that we're studying have relatively low mortality. So you would need to do huge trials to be able to be powered to show a reduction in mortality. And some of the devices or drugs you're testing are really not meant to reduce mortality. They're meant to improve quality of life, or prevent hospitalizations, or prevent restenosis, or other things that may not directly affect mortality. So you have to be able to choose the right endpoint. You have to be able to know how to measure it, how to collect it. And then that will often serve as your primary endpoint for the study. And the whole powering of the trial, and the number of patients that you need, will usually be based around your primary endpoint. So you have to have a good estimate of, one, what you think the rate of the primary endpoint will be. And that will depend on the endpoint that you've chosen, the measure itself, the risk of the patients you're enrolling, and how long you follow the patients for. Because ultimately it's going to be the number of adverse events in the trial that will determine how large the trial needs to be. So you'll come up with your primary endpoint. You'll come up with a lot of secondary endpoints that you want to measure, even if you're not powered for them, even if you're not powered to make a claim, a regulatory claim. If you're powered to show reduction in hospitalization, maybe you'll also want to be able to show improved quality of life. Maybe you'll also want to know that you can show improved left ventricular function. So if so, you've got to consider carefully what those parameters are going to look like in your control population, and you've got to measure them. If you're going to show improved left ventricular function, you've got to be able to routinely measure baseline and follow-up left ventricular function. And then you've got to measure it in a very accurate way. So you often can't just rely on the sites, which will read their own laboratories will read LV function data differently. There's a lot of inter-site variability, and even at one site, there's a lot of inter-reader variability. So we often will employ core laboratories, where all the data gets sent to do independent and unbiased reading to get rid of that variability between the sites. So you've now chosen your patient population, you've now chosen your endpoints. You then have to get your statisticians involved to power the trial. And so you've got to be realistic. And a lot of the reasons that trials fail is because investigators are unrealistic. They think the event rate in the control arm is going to be too high. Or they think their device or drug effect size is going to be too great. And very few drugs, devices, or therapies will reduce whatever you're going to measure by 80%. In this day and age where we've got good treatments for not all, but most therapies, probably 10 or 20% treatment effects are more realistic, not 80%. But 80% lets you run a small trial. And if that's negative, then you conclude we were unable to show an 80% reduction. But that's not very realistic. So you'd like to power your trial based on a realistic sample size, but then it gets very expensive. So ultimately, you power your trial, you determine what is accurate, and then you have to budget it. And when you budget it, then you'll find out whether it's within your budget or if it's not within your budget. And if it's not within your budget, then you've got to figure out ways to cut costs. And you can cut costs by reducing the number of patients, reducing the length of the follow-up, reducing some study quality metrics. Sometimes you can reduce something. Sometimes we over-engineer trials and we spend a lot of money, we waste a lot of money. We monitor things that don't need to be monitored. We collect data that doesn't really need to be collected. And all that costs money. We have inefficient startup activities, and that costs money by slowing down patient recruitment. So you've got to figure out how you can improve the efficiency of clinical trials. Ultimately, you get to a place where you've got a study design that's powered with an endpoint that you can afford. And then you've got to consider all the study processes to optimize the way the clinical trial is going to run, not from both a clinical trial point of view, getting sites trained, getting sites initiated, having them routinely screening for patients, operationally enrolling patients appropriately and according to regulations, filling out case report forms as the patients are enrolled, sending films to core laboratories or EKGs to core laboratories, whatever data that you're collecting, and overseeing that. Either by the study sponsor or by the contract research organization. And then you've got to have all the studies in place to make sure that you're optimizing the likelihood of your drug or device to be successful. So if I'm going to use a new stent, I've got to decide what pressure should it be implanted at. How far should it cover the lesions? Where do I implant the stent? What stent to artery ratio do I use? And then you've got to actively track the trial as it's enrolling to make sure the sites are actually following those directions. So what anticoagulants do you use? What antiplatelet drugs do you use? And are the patients are the sites following the inclusion and exclusion criteria for the patients? Or are they trying to take liberties to enroll more patients? So you've got to very actively manage a clinical trial to make sure that all of the study processes that you wrote in the protocol are being followed. So, and this just is, again, this was probably more than you wanted to hear, 5 or 10 minutes, but I could go on for another 30 minutes talking about all of the clinical trial details that you've got to oversee to make sure that it's going to run as efficiently and to give you the accurate results that you hope to get.
Yeah, this was great, and thank you for walking through the whole process, at least from your perspective as a researcher and a clinician. What are your thoughts about doing clinical trials in other countries, like outside the U.S. and combine? Like, is it better to strategize your trials where you have U.S. and OUS countries in the same study, or is it better to do separate trials?
That's a great. Depends a little bit what you want to accomplish. I mean, if you truly want your study to be generalizable, that is, that it applies to all peoples around the world, then you need to make sure that the study centers that are enrolling and their background practices, the background medical therapies that they do, and the way they practice intervention are included in the study. If you want to make sure that your trials doesn't apply only to white Caucasian men, you've got to make sure that you enroll enough women and you enroll enough other patient groups, whether they're blacks, Asian patients, Pacific Islanders, or whoever, because the trial results that you get are going to apply to the types of sites that enrolled. And a lot of that is based on geography, as well as the patient profile of the patients you actually enroll. So, for example, in Eastern Europe, many Eastern European centers do things very different than Western Europe. Western Europe tends to do things relatively similar, but into the United States, but in some respects quite differently. So, if I do want my trial to apply to not only the United States, but also to most of Europe, I'm going to want to have good representation across the United States, north, south, east, and west. And I'm going to want a number of sites, a good involvement of sites from both East and Western Europe. And then they may not apply to the Asia Pacific region, because the way things are practiced in Japan or China or South Korea are often different. The patients are often different. The coronary disease is different. There are different genetic polymorphisms. So if I included, 10 sites from China or from Japan, and then I can look at heterogeneity. Did the Japanese patients respond similarly to the way the American or the European patients responded? Then I can make some claims of generalizability. But if I didn't include those patients in my trial, then my trial will only apply to the limited geographies I did include. On the other hand, it's probably much more easy to control the trial if all you did was a United States trial. The patients are more uniform, the background procedures are more uniform. There's probably going to be less variability that may affect the results of. We in the United States, we tend to send PCI patients home now the day of the procedure for elective PCIs. In Europe, there's a financial incentive in many situations to keep patients in the hospital 4 or 5 days. So if duration of hospital stay is an important metric, then I can't really look at that and have it apply to a U.S. Population if I'm also enrolling a lot of European sites. So that's just one small example. So there's no right answer as to whether just a U.S. centric or a U.S. plus Western Europe or U.S. plus pan-European, or to truly make it global effort is more or less appropriate. And it really also depends on your budget. The more countries you do, the more expensive. In general, it makes it regulatory-wise much more complex. Every country has its own regulatory requirements that you have to track and be in compliance with. You need very large and very well-funded studies to truly be multinational.
So, what are your thoughts on using real world evidence and pragmatic clinical trials in complementing traditional randomized control trials in cardiology?
So there's a couple ways that people could use the term real world evidence. There's the concept of doing registry based randomized trials. For example, the way they've really perfected or popularized it, and almost perfected, I would say, in Sweden with the Sweetheart registry. And in Sweden, basically all patients that live in Sweden are enrolled in one or more registries, overall under the banner of Sweetheart. And there's PCI registries, there's readmission registries, there's heart failure registries, and they're all linked by unique patient identifiers. And that's all put into a national database system whether or not patients are in a clinical trial or not. And so many investigators in Sweden, some of whom are some of the best investigators in the world, have learned to use that registry-based system in which outcomes are also put into the database to run very inexpensive, large-scale randomized trials. For pennies on the dollar as to what we might spend in the United States. And I think there's tremendous power in doing that. You can tend to enroll randomized trials much faster and, again, much less expensively. Where I think that falls apart is that they're best designed to answer very simple questions with simple endpoints, such as all-cause mortality, because there's very little uncertainty in adjudicating all-cause mortality. If you've got a good national register that really you can believe that the patient has died, they're really registered in that administrative database as no longer alive, then you don't really need to adjudicate that. On the other hand, most of our trials have clinical events committees to adjudicate some very sophisticated endpoints. We still have trouble agreeing on what a myocardial infarction is, for example. What meets the criteria for a stroke? What is heart failure? And I'll come up with 3 or 4 different criteria. And each criteria, you can have 3 or 4 possibilities, and you got to have one met of every bucket before you can say it was heart failure. And so usually you have to you have to look at the detailed source documents to confirm that those criteria are present. And that's what a clinical events committee will do. And they'll spend anywhere from 15 to 30 minutes on an event, making sure that it actually meets your definition of that event. So again, I think that's one way to reduce costs if you do registry-based randomized trials, but they're really good for very, very simple endpoints, such as survival. The other use of real-world evidence, people now are talking about observational data. For example, we could also use Sweetheart. They'll have hundreds of thousands of patients that'll undergo revascularization, put into their databases, not being randomized to anything. And now you're just looking at, well, what are the outcomes of in those patients? And you can use observational data like that, which usually come from administrative databases, to look at the outcomes in men versus women, or the outcomes in Black versus white patients, or the outcomes of diabetic and non-diabetic patients. You can look at the outcomes of patients who happens to get PCI versus who happen to get bypass surgery. And you can then try to correct for the differences in those groups, because these aren't randomized questions you're asking. Okay. And so diabetic patients may be treated very differently than non-diabetic patients, not only in the use of stents or PCI or CABG, but also in their background medications. And they have different rates of renal insufficiency and different rates of polyneuropathy and gastropathy and other diseases that can affect their outcomes. And the beauty of randomization is it balances for all of those confounders. But when you're doing observational data, all you can do is try to collect as much of it as possible that you think might affect the outcomes, and then try to do multivariable analysis to adjust. But the problems with that is that we often don't collect all the important confounding measures. They're often not, observational databases are often not that complex or granular to have all that detail in it. They don't often have things like socioeconomic status or education level or home health care, which can truly affect outcomes in trials. Randomized trials, if you don't collect it, it'll balance out between the two arms. But if you're going to do observational data, diabetics and non-diabetics may have very different situations in that regard, for example. So I think there's a relatively limited role, in my opinion, for observational data for comparative health effectiveness outcomes. However, the beauty of observational data is it's relatively inexpensive. It probably costs 1/100th of what a randomized trial costs, and you can truly have big data. You can have hundreds of thousands or millions of records. Okay, so you would think there's got to be some utility in that. And I think there is utility in it. First of all, I think it's very useful to see how people are practicing, how many people are getting PCI versus bypass surgery. How often is intravascular ultrasound used during coronary interventional procedures? Okay, how often are patients being treated with a year of dual antiplatelet therapy after PCI compared to a month or 3 months or 6 months? So practice patterns is very, very useful. And I think it's also very useful to uncover low frequency safety events. So, for example, most of our, pivotal device trials are in the hundreds of patients size, sometimes 1,000 or two. Most pivotal drug studies are, in the thousands, sometimes 10,000 or so. But sometimes it might take hundreds of thousands or even a million patients to uncover a very rare association between a drug or a device and an adverse outcome. Maybe one in 100,000 balloons are going to rupture, but when it rupture, it's going to cause a catastrophic perforation and death. Okay, so you may be very lucky, and if you do a randomized trial of 1,000 patients, you may never detect that. You may never see it. But then once you treat 100,000 patients and it happens 2 or 3 times, that may be enough to recall that device and improve that device so that doesn't happen anymore. So I do think there's utility in large observational databases and also in generating hypotheses. So do comparative effectiveness outcomes analysis, but it really creates a hypothesis. And then if you come up with something important enough, gosh, I happen to note that diabetic patients that get treated with hydrochlorothalidone rather than Lasix look like they have less heart failure. And gosh, that data seems really strong in this database, even after I do all multivariable correction. Well, then do a randomized trial of those 2 agents in patients with diabetes with a heart failure endpoint. But it's really got to be hypothesis generating because you never can correct for all the confounders you could measure or that you don't. We call those unmeasured confounders.
Yeah. The other use case is the post-market clinical follow-up reports that are needed for EUMDR.
Right. Yeah, and that gets into it. I mean, for EUMDR, of course, you're mainly approving based on safety, right? You're not necessarily approving on effectiveness. And of course, you're approving based on safety, often in a limited number of patients, often much less than we require in the United States, which requires reasonable assurance of safety and effectiveness for medtech devices. So it's even more important in Europe, where devices in particular are put on the market with relatively little data, and so you really need those robust post-market surveillance efforts to make sure devices are safe, let alone whether they're effective or not.
I want to talk about like what are some of the key factors that lead to successful collaboration between study investigators such as yourselves, industry sponsored, and regulatory agencies. And I also want to understand like your process, like how do you manage disagreements with sponsors? Like you touched on, different aspects of the clinical trial design, and then if you change the levers, it changes the sample size, which means like you're spending more money, because you have to enroll more patients, and then there's things that you may need to as a clinician to justify or not justify but to support the hypothesis, but the sponsor may disagree, and kind of how do you navigate those discussions?
Sure. So on one hand, the minority of clinical trials are government sponsored, in particular NHLBI sponsored. And the beauty of that is that most clinicians or academicians' interests are aligned with the government, and that you're really trying to improve outcomes for patients. That's the only main goal. But the disadvantage of government-led trials is, one, there's a lot of bureaucracy, and two, they're often not resourced as highly as they should be, and so you often have to cut a lot of corners in that. So I've actually probably spent the majority of my career working with industry sponsors. And industry sponsors tend to be better resourced. The goals, though, of industry may or may not always be aligned with your goal as a clinician. I mean, my goal as a clinician, my primary goal, honestly, is not to help a sponsor succeed. My primary goal as a clinician, having gone to medical school, having devoted my life to taking care of patients, is to try to perform a clinical trial that's going to improve patient outcomes. However, I think that my goals should be aligned with industry goals because industry makes the tools, they make the drugs, they make the devices that I need to be successful in caring for my patients, that society needs to be able to help us improve outcomes for patients. And, in my estimation, if industry makes a drug or device or another tool that helps improve outcomes for patients, they should be rewarded for that. And they should be able to make a healthy profit for that. So I always see that my goals and industry goals should be aligned. And we're in an era now where everything is scrutinized and looked at very, very carefully by regulators, by society, by journals, there's no shortcuts. If industry primarily is trying to say, you'll have some industry, primarily startup companies, that may or may not be resourced as well, or maybe somewhat less ethical. And they'll try to cut corners, or they'll try to have endpoints that are clearly going to try to just make the trial positive. We really don't care about coming up with something that's going to be important for patients. Fortunately, there are not that many companies that are out there like that, but you always have to guard against it a little bit. The really good companies, the ones that are well established, the ones that are growing and are going to be around forever. You know who they are in pharma. It's Pfizer, it's Merck, it's Bristol Myers Squibb, it's AstraZeneca, and in medtech, it's Medtronic, it's Abbott, it's Boston Scientific, it's Johnson and Johnson, it's Edwards. You can go on and on. These companies really, I believe, our interests are aligned. And I do believe that in their R&D divisions, their number one goals are to try to come up with important findings that, yes, are going to promote their products, but they're going to promote their products because they improve outcomes for patients, and physicians will want to use them, will want to prescribe them because physicians who have only loyalty towards their patients believe that their patients' outcomes will be better. So rarely have I had a situation where I would say that I've butted heads with industry about important issues. Where it does become unavoidable sometimes is when costs get out of control. And, that's understandable. I'll say, look, I'll say to an industry sponsor, it's not reasonable to think that this device is going to reduce the treatment effect size by 50%. It's more likely, so you shouldn't power the trial for 50%. It's more likely 20%. So, okay, let's be optimistic. Let's power for 25%. But then the sponsor will say, no, that increases my sample size from 500 patients to 3,000 patients. And that's going to take me 3 more years to do and increase my budget, from $10 million to $45 million. And I can't afford to do that. Okay. So then you've got to work with industry. And if you're collaborative as the academician, and if industry is collaborative, understanding what you said is a real limitation, then hopefully you'll find a middle ground. And you usually can, but sometimes it's difficult, because the last thing I tell industry, whether it's an established company or whether it's a startup company, is you don't want to rush to a finish. You don't want to rush to just get your clinical trial done and it be negative, because then the startup company will usually go bankrupt, or the main established company will just then bury the therapy and move on to something else. So nobody wants that. But it can take very hard decision-making to get to a point where, you can both afford a clinical trial, and they're getting more and more expensive every day, and optimize the likelihood of it being successful within your budget.
Yeah. One of the things I've observed you do really well on the projects that I've worked with is like you're really integrated with the project team at the sponsor. Like you will like be very clear on what type of analysis you want to see and so on. Do you get pushback on that and like, oh, that's too much work, Dr. Stone, we can't do it, and kind of how does that dynamic work?
Yeah, there's two kinds of sponsors. There's one kind of and two kinds of investigators. One kind of sponsor, they want to design the trial, they want to run the trial, and then they just want to choose a PI to be a figurehead.
Hmm.
And frankly, yeah, come to an investigator meeting or two, and we'll let you present the data. And we're just going to give you all the data. We'll make the slides for you. We'll even write the paper for you. Okay. And some investigators might like that too because it's not that much work. But I think most investigators that are worth their salt don't want to do that. They want to be very, very involved in the process. And the best kind of partnerships, I think, work when it is a true collaboration between the sponsor and the investigator, where you ask the questions together, you choose the patient population together, you design the study together. And it can take hundreds of hours to do that, and you've got to be willing to put that time in. One piece that's often overlooked is the case report form. There's nothing more boring than designing a case report form, but there's also nothing more important. If you don't ask the questions on the case report form, the data doesn't exist because you can never get it later. You got to anticipate up front everything that you're ever going to want, and you're going to have to ask the question very accurately so you can collect it. And I end up, I'll spend 40 or 50 hours on case report form design, which is crazy because, again, it's incredibly tedious, but boy, does it pay dividends down the line. So then there'll be, and then the amount that you will then integrate with the sponsor on some of these tasks will depend a little bit on the expertise of the sponsor, right? You take, companies, for example, good med tech companies, they'll have their own integrated clinical trials organizations, and they may have run, 20 or 30 randomized trials before. So they'll have a lot of experience. Okay. So I've run about 150 randomized trials. So I have a lot of experience, and they can teach me things and I can teach them a lot of things. Sometimes with startups though, there'll be people who have never run a randomized trial. And they'll count on you for a lot. And you may really have to carry them along, tell them who else they need to hire, what other organizations they need to get, what CROs, regulatory consultants, statistical consultants, et cetera. So then your expertise in some ways is even more unnecessary. So again, you've got to, it'll really vary depending on the experience level and knowledge of the investigator and the sponsor.
Sure, that's great. Thank you so much for explaining that. I think I want to wrap up our conversation with a couple more questions or three more. I want to, you've been published in some of the leading journals like New England Journal of Medicine, JAMA. How do you do you have any suggestions or advice or guidance or techniques that you use to effectively collaborate with these editors, and then how do you go about managing revisions and rejections?
Yeah. So, in general, the time to start thinking about that issue is when you're designing the trial. Uh, I do know many, not all, but many of the editors of these journals. But if I submit something that's unworthy to them, they're not going to do me a favor and publish it. Okay. And everything is subject to peer review. And when you're talking about, the leading journals that we try to get into, which is usually the New England Journal, Lancet, and JAMA, you're talking they'll accept, 3 to 5% of original research manuscripts. So they're going to reject 19 out of 20 or more. So you've got to be the best of the best. And so I know what they're going to want. I know the quality that they're going to want. I know the screening logs that they're going to want. I know the power analysis that they're going to want and the statistical expertise that they're going to want. And the time to know all that is when you're planning the trial. Because if you didn't plan the trial right and you didn't collect all the information that they're going to want, they're going to say, nah, well, that, just doesn't meet our standards. But then hopefully, so it all goes hand in hand. And then hopefully, at the end of the trial, when you finally got the data in hand, journals like Most of the journals like relatively clear results. The best thing is to have a very, very positive study, right? In some of these guides, the second best thing is to have a very negative study. There used to be a big bias against negative publication, but now journal editors are trying to overcome that bias and trying to publish really well done negative trials because they also give you an important lesson. When it can be really difficult is when the message is nuanced and trials are borderline positive or borderline negative, or only subgroups are positive, and there's too much crossover, and so maybe the per protocol population is positive, but not the intention to treat is positive. And when you get too nuanced, sometimes it's too much for the major medical journals, which do want more of a clear message. And so then you end up in the cardiology specialty journals. So the bottom line is you do your best job on every publication. And I try to write the same quality and level of publication, whether it's meant for the New England Journal of Medicine or whether it's meant for, a very low impact journal. You try to do your best. You try to include all the elements that you think any peer reviewer would like to see, and then you try to get it in the best journal that you can.
Great. Thank you, Dr. Stone. Fully agree. And then, do you have any resources that you'd like to share with the audience today? Maybe it's books, but it could also. I know you're part of research that's kind of going into the books that are getting published, so you're kind of ahead of the publications. But I want to see, how do you keep yourself current with the industry and things that are going on in research?
Yeah, so keeping current, as you say, is critical. I mean, in my 30 to 35-year career, there's probably a handful of drugs that are still in use when I finished my fellowship. So every 10 years, probably 50% of all knowledge and practice turns over. So you've really got to stay current. There's a lot of ways to stay current. It starts with being around really smart people and really smart colleagues at your own institution and going to multidisciplinary conferences and listening to what they say. Not just think you know every answer for the rest of your career. And then going to meetings, reading journals. I'm not a big believer in books anymore, I must say. I haven't opened a book in more than a decade. By the time books are published, everything's out of date. So, but I also don't get any paper journals anymore. It's probably been a decade since I've opened a paper journal. So everything is online, everything's instantaneous, but you've got to devote the time to keeping up, and you've got to avail yourself of all of those resources and choose the 3 or 6 or 10 journals that you want to keep up with. You'll never be able to read them all, but choose the key articles that you're going to read. Try to scan as many as you can, and then choose the ones that you want to go in depth in and really read the article from beginning to end and try to understand the nuance, especially the important studies. Through that, through conferences, through networking, there's probably some role for social media too. If you can keep the positives and avoid the negatives, you'll keep up. And again, this is a very a collegial and collaborative infrastructure that we work in. So you can't be an island. You've got to work with the companies, know the leaders in the companies, work with your colleagues in your own hospital, other hospitals in your city, other cities in the United States and internationally, if you really want to be a true clinical trialist, thought leader. And a key aspect also is statistics. I'm not a statistician, but I've learned a lot of statistics over the years. Everything I know about clinical trials, which is a fair amount now, I've learned on the go, and I've learned by making mistakes and watching other people make mistakes. And you try, it's critical. You learn much, much more from mistakes than you do from successes, and you have to integrate that into your thinking. And you've got to, again, rely on other people who are a lot smarter than you, especially as you're starting out. Try to choose mentors, try to be around them as much as possible, learn everything you can, soak up every word they're going to say. And then if you're super serious about this field and you want to become a clinical trialist, there are several, advanced master's programs that you could take in clinical trials. that places like Harvard or Columbia and other selected centers put on, just in clinical trial design and statistical support and analysis. So you could spend a year, sometimes even two, taking those, and then you get a big jump start if you'll spend that at the early stage of your career.
Are there any like top 3 journals that you typically see yourself going back to, or even like statistical resources?
And not as much statistical resources. I mean, most statistical journals and resources are above the level of most clinicians. But most of the good medical and cardiology journals will publish some statistical articles. And those tend to be at the level for cardiologists to read. Stuart, for example, has really been an example of a world-class statistician who likes to translate statistics into plain English, and has written a very case-based articles on statistics. I've collaborated with him on a number of these that have been published anywhere from JACC to EHJ to the New England Journal of Medicine.
What's his name? Stuart. What's his last name?
Pocock, Pocock. And his name as a statistician is on many of the standard statistical tests that we actually use. So, I mean, he is a world-class statistician, but he also believes in working with, the numbnuts of the world like me that are not grounded in statistics to teach me what I need to know about how to use and interpret statistics, when to analyze recurrent events, when to use Poisson distributions versus negative binomial tests, how to use joint frailty models. I mean, things I never heard of, 20 years ago. I learned from people like him and others, and I've incorporated them into clinical trial design. But then, of course, to actually make it happen, then you need real statisticians that know how to work the statistics and work the numbers. So I know just enough to be dangerous.
Great. And then one final question, Dr. Stone. I want to talk about like what keeps you going. Like you are always on a mission on the next clinical study, like on podiums at the largest cardiology conferences around the world. What is it that keeps you going, and how do you want people to remember you?
Well, thank you for asking that. I mean, what keeps me going is number one, the thirst to try to address and answer important clinical questions that, when we apply the answers to patients, are going to improve their lives, make them live longer and make them live better. And medicine is unending. The physician, most physicians that are practitioners care for one patient at a time and try to do their best to try to make that individual have the best outcome they can. And I've tried to do the same, but I try to do it on a larger level, on a medical population level, if you will. So the results of my studies can be used by thousands or tens of thousands of physicians to improve the lives of tens of thousands of patients. And there's always more that we don't know, and there's always suffering out there, and there's always disease. And while we've markedly improved the outcomes for patients with cardiovascular disease just in the 35 to 40 years in which I've been training and practicing medicine, we've still got a long way to go. So that's what drives me more than anything. And then I would say is a thirst for knowledge. I do think I'm a true scientist at heart, having the ability and the opportunity to, be a small cog, and, being humble enough and grateful enough to being in a position to being able to help answer these questions as one of a large number of people on any given clinical trial, and one of an infrastructure of tens of thousands of researchers that are trying to improve outcomes for patients is truly gratifying, and very special part that's made my life worth living, frankly.
Great. Thank you so much, Dr. Stone, for your time. This was amazing.
Thank you, Kunal.
Thank you for listening to the Clinical Trial Podcast. For more exclusive clinical research content to get you to the top of your game, head on over to clinicaltrialpodcast.com.